all our thoughts, fears, crushes, memories. Everything that feels most us is, in the end, just patterns of electrical activity. Have you ever wondered what that pattern in your brain actually looks like in the exact moment you see something or think of one specific idea? If you could pull one of those patterns out of the skull and let it float in the dark, what shape would it have? When I tried to draw one of those patterns from real brain data, it looked strangely alive, more like a deep-sea creature made of light than a diagram of a brain. This paper is where it starts. 10 people lay in an fMRI scanner and watched over 1,000 tiny 3-second video clips of everyday life. For each little moment, we get the video itself and a whole brain response. Each clip is also described in rich detail: what objects are there, what kind of place it is, what actions are happening, and a full sentence capturing the little scene. So it's not just pixels, it's a catalog of tiny lived moments, all with their matching brain patterns. Here, they check that those patterns are stable. The colored regions are places where the brain reacts to the same clip in almost the same way every time. So each 3-second video leaves its own repeatable imprint across visual and parietal cortex, like a small, consistent fingerprint of that moment. Then, they ask what those imprints mean. They compare the brain patterns to different ways of describing the clips: just the objects, just the scene, the action, or the full sentence. Different regions care about different things. Some line up best with what's in the scene, some with what's being done, and broader areas respond in a way that fits the whole little story. So these aren't just blobs of activity, they're carrying structure about what the moment is. From all of these clips, I chose one. It's a short video of a child playing alone in a room, filmed with this slightly washed-out, old VHS home video look. It already feels less like something happening now and more like a faded childhood memory. For that one clip, the dataset gives a map of how strongly every point on the cortex responds, and how reliably it does that across repeats. I filtered out the vertices whose response pattern is most unique to this clip compared to all the others. Basically, the parts of the brain that don't just respond, but respond in a way that stands out against the background of all the other moments. This is what that pattern looks like when you turn it into a 3D shape. It ends up looking like a living creature, almost a jellyfish made of light, drifting in the dark. A thick, glowing cap where the cortex is busily processing the scene, and long, colored tendrils hinting at information being routed to other regions of the brain. Most of the computation in the brain happens in a very thin sheet called the cerebral cortex, a wrinkled gray surface wrapped around the brain, just a few millimeters thick, packed with neuron cell bodies and their dense forest of dendrites. Under that sheet is the white matter, long bundles of axons, like cables, connecting distant patches of cortex. You can think of it as thousands of little processors spread across the surface, all wired together by a tangled cable harness inside. In my visualization, the thick, cloudy heads of these jellyfish shapes correspond to that surface sheet, where this particular clip drives the most distinctive, clip-specific responses. I start streamlines there and let them creep along the folds of the cortex and down into a model of the white matter tracks. Where the response stands out more for this clip, the web becomes denser and brighter. As it becomes less specific, the strands thin out and fade into black. With these tentacles, there's one thing to keep in mind. They follow the white matter pathways, the long cables that link distant regions of the brain. From diffusion data, we can estimate where those bundles run in general, but we can't see the precise route a single electrical signal took in this exact moment. So in this visualization, I let the activity begin near the cortical surface, and I let the streamlines fade as they dive inward along those tracks. In a real brain, the wiring inside this jellyfish would be far more extensive and tangled than what you see here, a dense storm of fibers instead of a few clean threads. Even with those limits, the sculpture is still anchored in real data. The starting points come from actual fMRI responses to that child in a room video, and the paths follow real anatomical wiring. It isn't the full truth of what happened in that brain, but it might be the closest sketch we can draw right now. To understand what this kind of picture leaves out, it helps to look at the raw signal. Even with modern, millimeter-precision fMRI, the pattern of activity you see is already wildly complex. And that's only an indirect shadow of what's really going on. The true electrical chatter of billions of neurons is even more intricate, but we only pick up slower changes in blood oxygen. If both scans would come from people just watching a movie. On the left, the clip it could be quiet close-up of a dim room. On the right, it's a noisy street scene with cars and crowds rushing past. But when you project the activity onto the cortex and play it over time, you mostly just see a swirling, ever-changing mess of color. Can you tell, just by eye, which one is which? Probably not. The brain is always buzzing, and fMRI gives you this high-dimensional flicker of activity that's really hard to interpret directly. It's rarely just a few neat patches turning on while everything else stays dark. This paper's bold moments dataset takes a different approach. Instead of keeping that whole flickering movie for each 3-second clip, they use a model to condense the response into one cleaner pattern per video, essentially smoothing over time and over repetitions of the same clip. The modeling is more sophisticated than just taking a raw average, but the basic idea is that we throw away a lot of the frame-by-frame detail in exchange for a clearer, more stable picture of where the response tends to be strongest or most reliable. This is very useful if you just want a rough map of which parts of the brain are involved, regions that light up more for motion or faces or sounds, or that track certain objects or actions across many clips. But it mostly captures what is shared across repeats, the general fact that something is seen here or something is heard here. In my sculpture, I push that one step further by focusing on the vertices that are most unique to the child in a room clip. So the web you're seeing isn't just about generic functions like vision or sound, it's more like the particular imprint this one video leaves against the background of all the others. But it's still a frozen imprint, a still frame of the story, not the whole temporal sequence. It doesn't really tell you about the inner texture of the moment, what your attention lands on first, how the meaning of the scene slowly builds over those 3 seconds, what quiet feelings or associations bubble up when you watch that child in the room. A lot of that seems to depend on the fine-grained timing and interactions between regions, on how patterns rise, line up, and fall away, which mostly gets blurred when we compress everything into one static map. That's why many modern ideas about consciousness focus less on isolated hotspots and more on the evolving dynamics between regions, on how distant areas briefly fall into sync, drift out of sync, form and dissolve coalitions of activity across the brain. You can think of it like a constantly shifting piece of music, different instruments, different brain areas, coming in and out, locking together in rhythm, breaking apart again. That moving pattern in space and time is probably much closer to the thing we actually feel from the inside. From that perspective, these static bold moments maps, and my jellyfish sculpture built from them, are like long exposure photographs of fireworks. They show you where things tend to happen, but not the quick, complicated dance that might actually give rise to experience. In the next video, we'll stop freezing the brain into single, still frames and try to follow those dances themselves, tracing how experiences move through a hidden landscape of brain states and seeing what new shapes appear when we let time back in.